I am Dr. Giuseppe Ramirez from San Raffaele Hospital in Milan, Italy. Today I will briefly present to you the main features of our research entitled Histone-specific CD4 positive T-cell plasticity in active and quiescent systemic lupus erythematosus. This study focused on systemic lupus erythematosus, or SLE, which is a multi-organ autoimmune disease characterized by a broad variety of clinical manifestations. Dysfunctional processing of cell death debris, aberrant type 1 interferon responses, and deregulated adaptive humoral immunity constitute hallmarks of SLE, while less is known about the role of T cells. CD4 positive T cells indeed coordinate antigen-specific immune responses in physiology and disease. Evidence in the literature supports the existence of autoreactive CD4 positive T cells in patients with SLE. These cells likely play a key role in orchestrating and maintaining SLE-specific autoimmune responses. Therefore, we reason that characterizing autoantigen-specific CD4 positive T cell subpopulations would have provided crucial information on mechanisms occurring deep in the pathogenesis of SLE. We focused on HLA-DRB1 restricted responses and used HLA tetramers bound to K peptides autoepitopes for direct ex vivo visualization of antigen-specific CD4 positive T cell in the circulating blood through flow cytometry. Antigen-specific T cells were also characterized in terms of polarization and differentiation stage, and their plastic variation were assessed during distinct phases of the disease. Suitable HLA peptide couples were defined based on the serological profile and HLA-DRB1 allele frequency of a well-characterized cohort of patients with systemic lupus erythematosus. In particular, there were 185 patients. Anti-double-stranded DNA antibodies were the most represented serotypes among SLE-specific antibodies. Therefore, we focused on anti-chromatin immunity and chose histones as the most reasonable protein target antigens for pathogenic antigen-specific T cells. HLA-DRB1 0301, which is a known risk allele for SLE, and HLA-DRB1 1101 were particularly frequent in our cohort and were selected for the following assays. In silico analysis, along with literature review, were performed to identify most promising histone peptide candidates to bind to HLA-DRB1 alleles. Flow cytometry studies were performed by comparing 32 patients with SLE, 11 patients with Takayasu arteritis, a T-cell mediated inflammatory disease clinically unrelated to SLE, and 10 healthy controls. Histone-specific T cells were selectively detected in patients with SLE, while total T cell population had negligible differences among groups. Histone-specific T cells were also correlated with a positive anti-DNA profile. Ex vivo observations were also corroborated by ancillary in vitro assays where peripheral mononuclear cells from patients with SLE and healthy controls were stimulated with the study peptides and characterized in terms of cytokine release and activation marker expression. We also compared antigen-specific T cell counts among patients with active versus quiescent disease, and histone-specific T cells were lower during active disease. This finding was particularly evident for effector memory and T regulatory cells. We hypothesized that these findings might be explained by migration of effector memory T cells into inflamed tissues during active disease, along with defective regulation of the inflammatory response due to numerical and possibly functional deficit of Tregs. Interestingly, circulating histone-specific T cells were also higher in patients under immune suppression. We believe that our observations might be useful for the development of novel molecularly based diagnostic and therapeutic tools for precision medicine in SLE.